Good morning, everyone. Welcome to the module on functional MRI. Today we'll specifically be focusing on the bold signal. We're going to break down the physiological basis of how exactly we capture brain activity. So a quick revision on the MRI signal. The signal starts with protons or spins, which precess around their own axes and around the axes of the static magnetic field, which they call B0 in the scientific literature. We use a radio frequency pulse to essentially knock the spin out of alignment. Then, once it is out of alignment, the spin relaxes to its original low energy state, and that releases energy, and a radio frequency receiver measures that energy that is sent out during this precession and relaxation, and that allows us to create images. So the bold signal we'll be talking about is fundamentally just about blood. So we're going to look at the physiology of hemoglobin. What hemoglobin does normally is that it carries oxygen around the body. Here we differentiate between oxygenated hemoglobin and deoxygenated hemoglobin, which, as the name suggests, is respectively when hemoglobin carries oxygen and when hemoglobin does not carry oxygen. It's its two states. When neurons are active, what they do is consume this oxygen from hemoglobin. So they convert oxygenated hemoglobin to deoxygenated hemoglobin when active. And building on that, there is more deoxygenated hemoglobin if there are more active neurons. So if a particular part of the brain is more active, you will have more deoxygenated hemoglobin in the blood vessels in that area. So in response to this depletion, the body senses areas of low hemoglobin through its various mechanisms, and in response, increases the blood flow. The mechanism behind this is called the hemodynamic response function, and we'll build on that in a bit. So now we're going to specifically focus on the bold signal. The bold signal is a physiological response. It is the physiological response that we just discussed. It is a change in the T2 star weighted MRI signal. The T2 star, as compared to the T2 MRI signal, is a change that occurs because of local variations in the ratio of the magnetic, so deoxygenated hemoglobin, to non-magnetic or oxygenated hemoglobin. And this ratio, or the difference between them, serves as an indirect measure of neural activity. And that is the bold signal. What bold stands for is very simple. It is the blood oxygenation level dependent imaging. So it's just as the name suggests. So just to clarify the magnetic difference between oxygenated hemoglobin and deoxygenated hemoglobin. Deoxygenated hemoglobin is paramagnetic, which means, in the simplest terms, that it does have its own magnetic property. And introduces inhomogeneity, which is differences, local differences. In like the magnetic field. On the other hand, oxygenated hemoglobin is weakly diamagnetic. So for the purposes of imaging, there's no magnetic field, no magnetic properties or field. And so it has a little effect on the signal of the MRI. The key concept that we need to understand is that both of these have different magnetic properties and thus different effects on the decay of the MRI signal, specifically the T2 star signal. Okay, so let's discuss the hemodynamic response function. The easiest way to understand this is to look at a very simple graph. So just a quick definition, the hemodynamic response function is the typical time course of the bold signal in response to a brief stimulus. So this brief stimulus can be whatever. Often in studies they use tapping of fingers, so tapping of your index and thumb. It could be clapping of the hand or anything that is likely to cause a stimulus in the brain. So here we can see at time 0, we see that we have the baseline here. So before time 0, actually, we have the baseline here. This is the balance between oxygenated and deoxygenated hemoglobin. So at time 0, let's assume that we asked the patient or the subject to clap. We see an initial dip in the bold response, which is where oxygen is consumed. So oxygen consumed. And that means we have more deoxygenated hemoglobin, so the MRI signal decreases. Then the signal rises sharply to this peak here. And this is where the oxygenated hemoglobin rushes into the area. This is often an overcompensation. As you can see, it goes very high. Overcompensation. Then after the stimulus ends, so this period here is where the signal drops, and then we see a post-stimulus undershoot, which is under the original baseline, which is the dotted line here. And the reason this undershoot occurs is not entirely clear in the literature yet. But after this, it just returns to its resting state again until the next stimulus. So just to cover the overall physiology, what is the cellular mechanism responsible for this bold signal, for the hemodynamic response function? And the answer is astrocyte metabolism. The process starts when an action potential, so the electrical signals in a neuron, releases glutamate into the synaptic cleft. So the synaptic cleft is the area between two neurons. Imagine that a course of neurons is a chain, and the links between each of the little chain pieces is what we call the synaptic cleft. There's a diagram on the next slide in which I'll be able to show you I'll show you on the diagram. So the process starts when the action potential releases glutamate, which is the neurotransmitter, into the synaptic cleft. The astrocytes, which are cells beside the synaptic cleft, they reuptake the glutamate back out of the cleft for recycling afterwards. And this reuptake requires energy. Using this energy, it triggers glucose metabolism, which is where the energy comes from. And glucose metabolism requires oxygen uptake from hemoglobin. And that basically, it's this astrocyte function that is the basis of the bold signal. The bold signal measures this metabolic demand, so the uptake of, so the use of glucose, of the active neurons and the supporting astrocytes. And importantly, something that we really need to understand is that it is not the direct action potential of the neurons that we're measuring. It is just the metabolic demand. Okay, so let's recap that metabolic chain using this diagram to visualize what's going on. When neuron 1 here, let's use purple. When neuron 1 here has an action potential, when it's firing, it releases glutamate into this area. So this is glutamate. Glutamate is released into this synaptic cleft. The astrocyte, which is this green cell here, this green one, carries out the uptake, the reuptake of that glutamate so it can be reused later. And this specific action, so these green arrows here, requires energy. And this pulls oxygen out of the surrounding blood supply. And this consumption of oxygen is what causes the initial magnetic shift that the bold signal measures, completing the circuit from thought to signal.